A single drop. That is all it takes. One drop of pond water pulled from that murky green puddle at your feet contains an entire universe. Not metaphor, not exaggeration. An actual thriving ecosystem where creatures hunt, fight, regenerate from death, and survive conditions that would kill you in seconds. Most people walk past ponds every day. They see scum, algae, nothing worth a second look. But beneath that surface film lives a microscopic battlefield where the laws of physics bend and biology gets weird. Creatures with no brain that can learn. Animals that have not had sex in 25 million years. Organisms that can survive being frozen at minus 272 degrees Celsius, boiled alive, blasted with radiation 6,000 times stronger than what kills humans, and shot into space. All of this packed into bodies smaller than the period at the end of this sentence. Scientists discovered most of these organisms 300 years ago with primitive lenses. Anton van Leeuwenhoek ground his own glass and peered into rainwater collected from his roof gutters in 1676. He saw what he called animalcules, tiny animals, living things so small they defied belief. The Royal Society in London thought he was lying until they built their own microscopes and confirmed his claims. 3 centuries of research later, and we are still discovering new species in pond water, still finding abilities that should not exist. Today, you're getting 7 of the most jaw-dropping creatures swimming in that drop right now. Not the common ones you have seen in textbooks, the ones that make biologists question everything. If you value documented microscopic life and real biological research like this, subscribe. It is the best way to support our work. Also, comment from which city you are hearing this from. This script is researched and factual using sources from peer-reviewed journals published in PMC's open access database, including a 2022 paper from the Journal of Integrative Neuroscience on paramecium behavior, and a 2019 paper from the Journal eLife on tardigrade survival proteins, plus extensive documentation from Nikon's Microcosm U laboratory imaging database and UC San Diego's tardigrade research published in October 2019. These are not Wikipedia summaries. These are primary scientific sources from actual research labs. Now back to the information. You are looking at one of the largest single-celled organisms on Earth. Not largest by some microscopic standard, actually large enough to see with your naked eye if the light hits right. Stentor coralis stretches up to 2 millimeters long when fully extended. That is 80 times longer than most single cells. It earned its name from a Greek herald in the Trojan War whose voice could boom as loud as 50 men. Look at the organism under a microscope and you will see why. That trumpet shape, that wide bell opening. Abraham Tremblay first described it in 1744. He thought it was a hydra. He was wrong. This is a ciliate. Single cell, no brain. But watch what it does. The rim of that trumpet contains approximately 250 membranelles. Each membranelle is a cluster of cilia fused together into what scientists call supercilia. They beat in coordinated waves, creating a vortex that sucks bacteria and small protozoans straight into its mouth. The cytostom. Food gets pulled down a gullet and packaged into vacuoles where enzymes digest it. All of this happens in an organism with no nervous system, just one cell making decisions. The color gives it away. That blue-green pigment is stentorin. Scientists publishing in PMC documented in 2019 that stentorin may protect the organism from light damage. When you expose Stentor coeruleus to bright light, it contracts into a ball and hides. That is photodispersal behavior, a single cell running from light. No eyes, no brain, just chemical gradients and ion channels making it move. Research from the University of Oxford shows Stentor can learn, not learn like memorize. Learn like adapt behavior through repeated trials. Scientists put Stentor in tubes, timed how fast they escaped. Over multiple runs, the organisms got faster. They changed their behavior based on experience. A 2 millimeter single cell learning from mistakes. The nucleus looks like a string of beads, macronuclear nodes connected in series, sometimes 10 nodes, sometimes 20. This structure controls daily metabolic functions but does not reproduce. For reproduction, Stentor uses a separate micronucleus. When conditions turn bad, it can form a cyst. When you cut Stentor in half, each piece regenerates a complete organism within days. The head grows a foot, the foot grows a head, cut it into 5 pieces, and you get 5 new Stentor. Thomas Hunt Morgan studied this in 1900. He found that pieces as small as 1/27th of the original cell could regenerate if they contained part of the macronucleus. Stentor polymorphus takes this further. It hosts symbiotic algae, Chlorella vulgaris, green algae living inside the cell. Stentor provides shelter. Algae provide food through photosynthesis and absorb the Stentor's metabolic waste. 2 organisms in one body. Fossil evidence shows Stentor has looked virtually identical for millions of years. The form works, the mechanics work, evolution left it alone. Scientists at multiple institutions, including research documented in Current Biology, confirmed Stentor can contract its body to 1/6 its total size using kelomer fibers made of microtubules and M bands made of microfilaments. These cytoskeletal fibers run underneath the clear bands on the cell surface. When calcium ions flood the system, the fibers contract. The entire organism compresses into a tight ball in milliseconds. That contractile vacuole on its left side pumps out excess water. Fresh water keeps flooding in through osmosis. The vacuole works constantly expelling it, or the cell would explode. Stentor coeruleus does not age. Daniel Martinez at Pomona College studied related organisms and found they show no senescence. They do not get old and die. They just keep dividing. 1 becomes 2, 2 become 4. Barring predation or environmental catastrophe, they could theoretically live forever. But recent research from 2022 published in the Journal of Integrative Neuroscience reveals something wild. Scientists call paramecium a swimming neuron. It has no nervous system, no neurons, just one cell. But it generates calcium-based action potentials exactly like nerve cells do. When something touches the front of a paramecium, mechanoreceptors in the membrane open calcium channels. Calcium floods in, the cell depolarizes. That electrical signal travels through the cytoplasm. Within milliseconds, every one of the 4,000 cilia covering its body reverses direction. The paramecium swims backward, stops, rotates, then swims forward in a new direction. Scientists call this the avoiding reaction, one cell computing an escape route through electrochemistry. Research published in PMC in 2021 using high-speed cameras at 500 frames per second measured the ciliary beat frequency. Body cilia beat between 15 and 45 hertz, but the oral cilia around the mouth beat at nearly double that rate, 80 to 90 beats per second. These oral cilia create a feeding current that draws bacteria toward the cytostome. The organism can move at speeds up to 2 millimeters per second. That sounds slow until you scale it. If a paramecium were the size of a human, it would swim 4 times faster than an Olympic gold medalist. The cilia beat in metachronal waves, like stadium fans doing the wave, each cilium slightly out of phase with its neighbor. This creates the appearance of waves rippling across the cell surface. Studies using magnetic levitation at the National High Magnetic Field Laboratory in Tallahassee, Florida, subjected paramecium to simulated zero gravity. Scientists found that organisms maintained straight swimming trajectories even without gravitational orientation. They relied purely on chemical gradients and mechanoreception. Inside that single cell sit 2 nuclei. The macronucleus, shaped like a kidney bean, contains the working copies of genes. The micronucleus, much smaller, holds the germline genetic material. During conjugation, 2 paramecium attach at their oral surfaces and exchange micronuclear material. This is sex, not reproduction. No new individuals form, just genetic exchange. After conjugation, they separate, and each divides by binary fission, producing genetically diverse offspring. Research shows paramecium can be trained. A 2006 study published in Experimental Gerontology applied 6.5 volt electric currents to paramecium and measured their response to brightness levels. The organisms learned to discriminate between different light intensities. Single cells modifying behavior through what appears to be cellular memory, not neurons, not synapses, just biochemical pathways encoding information. The pellicle covering the cell shows parallel striations running around the body. Under electron microscopy, these resolve into rows of basal bodies, each anchoring a cilium. The invariant field at the cell's anterior contains cortical units with 2 cilia each. The posterior contains units with single cilia. This precise geometric arrangement never varies within a species. Paramecium tetraurelia has a fully sequenced genome. Scientists found evidence for 3 whole genome duplications in its evolutionary history. The organism rewrites its own genetic code. Some stop codons get reassigned to code for amino acids. UAG and UAA, which normally signal stop in most organisms, code for glutamic acid in paramecium. Paramecium expends more than half its energy on locomotion. That ciliary beating burns through ATP constantly. Scientists measured the efficiency at less than 1%. Terrible by engineering standards, but optimal for organisms with cilia that short. The contractile vacuoles pulse every 10 to 15 seconds. Each cycle collects excess water from the cytoplasm through a network of radiating canals, then expels it through a pore in the pellicle. Without this system, osmotic pressure would rupture the cell within minutes. 8 stubby legs tipped with claws, barrel-shaped body. They move like bears lumbering through moss. Most tardigrades measure between 100 to 500 micrometers. Some species reach 1.5 millimeters, large enough to see as tiny white specks in bright light. But size is not what makes them famous. Tardigrades survive. Period. Scientists at the University of Wyoming published Research in Communications Biology in November 2022 documenting how tardigrades withstand complete dehydration. They discovered a sugar called trehalose works with a tardigrade-specific protein called Cas-D. Together, these molecules stabilize cells during water loss. When a pond dries up, tardigrades enter cryptobiosis. They pull in their legs, retract their heads, shrivel into a structure called a ton. Metabolism drops to 0.01% of normal. They can stay in this state for years, decades. A 2021 study published in Hydrobiologia reported bidelloid rotifers surviving 24,000 years frozen in Siberian permafrost. Tardigrades likely match or exceed this. Temperature does not kill them. Research documented in multiple papers shows survival at minus 272.8 degrees Celsius. That is 1 degree above absolute zero. The temperature where atoms theoretically stop moving. Heat them to 151 degrees Celsius, and some survive. For short periods, radiation 6,000 times stronger than a lethal human dose fails to kill them. A protein called DISUP damage suppressor binds to their DNA and shields it from ionizing radiation. Research from UC San Diego published in eLife in October 2019 discovered DISUP forms a protective cloud around chromatin. When hydroxyl radicals attack the DNA, DSOP absorbs the damage. Scientists think this adaptation evolved for surviving desiccation, not radiation. When moss dries, hydroxyl radicals form. DSUP protects against those. The radiation resistance is a side effect. Tardigrades went to space. The European Space Agency launched thousands into orbit in 2007. They exposed some to vacuum and cosmic radiation. Others got shielded from radiation but exposed to vacuum. 68% of the vacuum-only group survived. Some even laid eggs in orbit. The eggs hatched. The unshielded group had higher mortality, but some survived complete exposure to space. The 2019 Israeli lunar lander carrying tardigrades crashed on the moon. Scientists debate whether they survived impact. Research published in Frontiers in Microbiology calculated tardigrades can withstand impacts up to 900 meters per second and shock pressures of 1.14 gigapascals. Anything faster turns them to mush. Meteorite impacts typically exceed those pressures, so the lunar tardigrades probably died, but the possibility remains open. Tardigrades can repair double-strand DNA breaks. The same protein pathways that let beetalloid rotifers survive radiation work in tardigrades. When gamma rays shred their chromosomes, they reconstruct them. Not perfectly. Some mutations accumulate, but enough fidelity to maintain life. They eat by piercing cells. A tube-like mouth contains needle structures called stylates. They stab into plant cells, algae, or fungus and vacuum out the cytoplasm. Some species eat other tardigrades or rotifers. All 4 of their front legs have claws. The 3 rear pairs anchor them while hunting. Under electron microscopy, the claws resolve into curved hooks made of cuticle. Scientists have identified roughly 1,300 tardigrade species. The true number probably exceeds 3,000. They live everywhere water exists. Mountain streams at 6,000 meters elevation, ocean trenches 4,700 meters deep. Antarctic ice sheets, temporary puddles, roof gutters. They need only a thin film of water coating their bodies. When that dries, they tune and wait. Tardigrade eggs can survive desiccation. Older embryos survive better than young ones. The chitinous shell protects them, but survival rates vary by species and humidity. Males are smaller and less common than females in many species. Some populations contain no males at all. Females reproduce by parthenogenesis. Eggs hatch into genetic clones. Tardigrades have simple nervous systems. A brain ganglion in the head connects to a ventral nerve cord. This sends signals to muscles in each leg segment. The complete neural connectum has not been mapped, but work continues. They have no respiratory system, no circulatory system. Oxygen diffuses through their cuticle directly into cells. Nutrients move by muscle contractions, squeezing the body cavity. The gut runs straight from mouth to anus. Extremely simple. Extremely effective. Fossil evidence places tardigrades at 500 million years old. They survived all 5 mass extinctions, survived climate shifts that killed 90% of other species. Survived because they can shut down, wait, restart when conditions improve. That ability keeps them alive through everything. November 2022, an extensive documentation from the National High Magnetic Field Laboratories studies on organism behavior in simulated zero gravity. These are peer-reviewed published papers, not internet summaries. Now back to the information. Anton van Leeuwenhoek found them living in his roof gutters in 1703. He discovered they could survive desiccation and come back to life when water returned. He called them animalcules, and the name stuck for 200 years. Bedelloid rotifers measure between 150 and 700 micrometers. Most are slightly too small to see without magnification, but appear as tiny white dots under bright light. The corona around their head looks like a spinning wheel. 2 rings of cilia beat in opposite directions, creating a vortex that draws food toward the mouth. Rota means wheel in Latin. That is where the name comes from. But here is what makes bedelloids impossible. No males exist. Scientists have examined approximately 500,000 bedelloid specimens over 300 years. Laboratory cultures, wild populations, Antarctic ice cores, sediment samples from every continent. Not one male has ever been confirmed. One report from 1923 described seeing a possible male among thousands of Rotaria rotatoria beneath frozen lake ice in Denmark. But when scientists returned to that lake in January 2019, they found no males and no ice. The winter was one of the warmest on record. That single observation remains unconfirmed and disputed. Bedelloids reproduce exclusively by parthenogenesis. Females lay eggs that hatch into genetic copies of themselves. No fertilization, no sex, no males. This has continued for at least 25 million years based on fossil evidence. Some estimates push it to 40 million years or more based on molecular clock analysis. Every rule in evolutionary biology says this should not work. Sex exists to purge bad mutations and generate genetic diversity. Without it, species accumulate damage and go extinct within a few million years. Bedelloids ignore that rule. They diversified into more than 450 species. They thrive in fresh water everywhere on Earth. Research published in 2020 and cited across multiple papers found evidence for genetic exchange between bedelloid individuals. Scientists studying Adenata vaga discovered the species might perform a non-reductional meiosis-like process during egg formation. Homologous chromosomes pair up and exchange material without actually having sex. This could explain how they avoid extinction. But the mechanism remains controversial. Many researchers dispute it. The genomes tell a weird story. Bedelloids have 2 or more divergent copies of every gene. Each copy sits on a different chromosome. Analysis shows these copies diverged millions of years ago. If bedelloids reproduced sexually, the copies would homogenize. They would blend back together through recombination. They do not. Each copy evolves independently. 4 copies of the heat shock protein HSP82 exist. All different, all functional, all on separate chromosomes. That pattern appears throughout the genome. Bedelloids also steal genes from other organisms. Horizontal gene transfer. About 10% of their DNA comes from bacteria, fungi, plants, and other sources. Scientists documented in 2025 that bedelloids copy dozens of antibiotic recipes from bacteria. The rotifers now use these antibiotics to fight off their own diseases. When you cut a bedelloid in half, both pieces regenerate if they contain part of the organism. The head end grows a foot, the foot end grows a head. Most species are oviparous. They lay eggs. Eggs measure 50 to 100 micrometers. Some species are viviparous. They give birth to live young. The young emerge fully formed, carrying their own eggs inside them. If bedelloids enter in hydrobiosis when water vanishes, they contract into a tun state, lose 95% of their body water, metabolism stops. In this state, they survive freezing at minus 80 degrees Celsius for more than 6 years. They survived liquid nitrogen at minus 96 degrees Celsius for weeks. Liquid helium at minus 269 degrees Celsius for hours. Ionizing radiation that would kill most organisms barely affects them. Like tardigrades, they can repair DNA double-strand breaks. When radiation shatters their chromosomes, they fix them. The June 2021 study published in multiple journals documented a bedelloid rotifer retrieved from Arctic permafrost that reanimated after 24,000 years. Radiocarbon dating confirmed the age. The rotifer extended from its tun state within minutes of adding water. It started eating bacteria. It reproduced. 24 millennia frozen solid, still alive. Bedelloids live in ephemeral habitats, puddles that dry weekly. Moss that bakes in summer sun. Film of water and lichen growing on Antarctic rocks. When conditions worsen, they tun. When conditions improve, they wake. This strategy works for 500 million years. They eat bacteria, fungal spores, algae, and other microscopic detritus. The corona sweeps food into the mastax. That is a muscular pharynx containing trophy. The trophy are 7 hard pieces that grind food like jaws. Under microscopy, the trophy look like tiny gears meshing together. Each species has distinct trophy shapes. Scientists use these shapes for identification because living bedelloids contract when disturbed, hiding most other features. Preserved specimens become shapeless blobs. You can only identify them when alive and active. Bedelloids crawl like inchworms. They extend the head, attach with the corona, release the foot, pull the body forward. Attach the foot, release the head. They also swim freely using the coronal cilia. Movement happens at around 1 body length per second. Not fast, but effective for their scale. 40 species live in Antarctica, documented in a 2015 paper in Hydrobiologia. They survive in meltwater pools, subglacial lakes, and wet moss in the dry valleys. Some species appear endemic to Antarctic environments. Others match species found worldwide, suggesting recent transport on wind or birds. The Antarctic populations endure winter temperatures that freeze solid every drop of liquid water. They tune and wait for summer thaw. Climate change threatens beteloids, not through temperature directly, through habitat disruption. As temporary pools dry faster and permanent ponds grow more frequent, beteloid populations decline. They evolve for ephemeral water. Permanent water hosts, predators, and competitors that outcompete beteloids in stable environments. Research shows beteloid diversity correlates with habitat instability. He thought they were plants. He cut one in half. Both halves regenerated into complete organisms within days. Plants do not do that. Animals do not do that. Tremblay had found something that broke all rules. He published his findings in 1744. The book described regeneration, budding reproduction, and feeding behavior in detail. Carl Linnaeus named the genus Hydra in 1758 after the many-headed Greek monster that grew 2 heads for every one Heracles cut off. The analogy fits. Hydra measures up to 30 millimeters long, but most species stay around 10 millimeters. That makes them barely visible without magnification. They attach to submerged objects using a basal disc at one end. 5 to 12 tentacles ring the mouth at the other end. When something small touches a tentacle, the hydra strikes. Each tentacle contains thousands of cnidocytes. These are stinging cells that fire nematocysts. A nematocyst is a coiled thread with barbs and toxins. When triggered, it explodes outward at accelerations exceeding 5 million g. That is one of the fastest movements in biology. The barbed thread pierces prey. Toxins paralyze it. The tentacle curls and brings the prey to the mouth. Hydra eat water fleas, insect larvae, small worms, and other microorganisms. Some species eat other hydra. After feeding, the tentacles contract and the prey disappears into the gastric cavity where enzymes digest it. Undigested material exits through the mouth, same opening for eating and excretion. Hydra has 2 cell layers, the outer epidermis and inner gastrodermis separated by a gel called mesoglea. These layers contain only 3 stem cell populations, but they generate all the differentiated cell types the organism needs. The stem cells divide constantly. A single hydra contains 50,000 to 100,000 cells total. Despite this simplicity, hydra regenerates from almost nothing. Cut one in half. Both halves regenerate. Cut it into 5 pieces. 5 hydras form. Cut it into 20 pieces. Every piece larger than 1/50th the original size regenerates if it contains stem cells. Scientists dissociated hydra into individual cells. Let the cells reaggregate into a clump and watch complete organisms reform from the aggregate. The cells sort themselves. Epithelial cells migrate to form layers. Endodermal cells move actively and organize the structure. Patterning processes create heads and feet. Within days, you have a functional hydra from a soup of cells. The molecular mechanisms involve key signaling gradients, but the details remain partially mysterious. Hydra reproduce asexually by budding. When food is plentiful, a bud forms on the body column. The bud develops a mouth and tentacles. When mature, it pinches off and becomes independent. A well-fed hydra buds every 2 to 4 days. Under ideal conditions, one hydra becomes a population of hundreds in weeks. Sexual reproduction happens when conditions worsen. Some species develop testes, others develop ovaries, some are hermaphroditic with both. Sperm release into water and swim to eggs in other individuals. Fertilized eggs secrete protective coatings and can survive harsh conditions. When the adult dies, the eggs remain dormant until better times. Research published in 1998 by Daniel Martinez at Pomona College claimed hydra do not age. They do not senesce. The paper documented hydra maintained in culture for 4 years with no decline in reproduction or survival rates. Later, work confirmed this. Hydra stem cells divide constantly and replace damaged or old cells. The entire organism turns over completely every 20 days. Nothing ages because nothing stays. Every cell is young. Predators eat hydra. The flatworm Microstomum linear specializes in hydra predation. Some species of coleps attack hydra in groups, tearing tentacles first. Fish eat them. Insects eat them. Hydra defend with nematocysts, but small size makes them vulnerable. Hydra viridissima looks green because it hosts symbiotic algae. Chlorella algae live inside endodermal cells. The hydra provides shelter and carbon dioxide. The algae photosynthesize and share sugars with the host. Both benefit. This mutualism lets Hydra viridissima survive in nutrient-poor water where other hydra starve. Hydra has neurons and a nerve net but no brain. Sensory neurons detect light, touch, and chemicals. Motor neurons trigger contraction. The nerve net connects them. Despite lacking centralization, the net coordinates complex behaviors. Hydra orient toward food. Avoid bright light. Contract when threatened. Move by looping. They bend over, attach tentacles to substrate, release the basal disc, and flip the body forward. Repeated looping moves them centimeters per hour. Fossil Cnidarians appear 700 million years ago. Hydra belong to this ancient lineage. Their simple body plan has not changed much. 2 cell layers, radial symmetry, tentacles, nematocysts. It worked then. It works now. Modern research uses Hydra to study regeneration, stem cell biology, and aging. The organisms are easy to culture. They reproduce quickly. Their transparency makes imaging simple. Genetic tools including CRISPR work in Hydra. Scientists have created transgenic lines expressing fluorescent proteins in specific cell types. This allows real-time imaging of regeneration and development. Understanding Hydra regeneration could inform human medicine. Tissue engineering, wound healing, age-related decline. The mechanisms are conserved enough that insights from Hydra sometimes apply to mammals. Not directly, but close enough to matter. Carl Linnaeus named it Hydra convallaria in 1758. Johann Rosel renamed it Vorticella convallaria in 1767. The name comes from vortex, whirlpool. Watch the cilia and you will see why. Vorticella has a bell-shaped body 30 to 40 micrometers in diameter when contracted. The stalk reaches 100 micrometers long. Some species like Vorticella campanula stretch the bell to 160 micrometers and the stalk to 4,150 micrometers. That is 4.1 millimeters. Visible without a microscope under bright light. The organism lives attached to substrates, aquatic plants, rocks, submerged wood, even other organisms. The stalk anchors it in place. Inside that stalk runs a spasmoneme. This is a contractile fibril that shortens when stimulated. Calcium ions flood in, the spasmoneme contracts. The stalk coils like a spring. The bell body jerks backward at speeds up to 90 millimeters per second. The whole movement takes milliseconds. Research published in PMC in 2018 calculated the spasmoneme generates tension between 10 and 100 nanonewtons powered purely by calcium binding. Per unit mass, it produces more power than a car engine. The bell has 2 bands of cilia around the peristome, the mouth-like structure. Inner cilia beat to draw water and food particles toward the cell. Outer cilia filter incoming material. Bacteria get swept into the cytopharynx. From there, food vacuoles form and digest the bacteria with enzymes. Waste exits through the same opening. Watch a feeding Vorticella and you see the vortex. A visible current spiraling toward the mouth, pulling in bacteria from hundreds of micrometers away. The flow velocity reaches 360 micrometers per second, at the peristome measured in laboratory studies with particle tracking. Vorticella reproduces by longitudinal binary fission. Unlike most ciliates, which divide transversely, Vorticella splits lengthwise along the oral-aboral axis. The process is unequal. One daughter keeps the original stalk and bell. The other grows temporary cilia at the aboral end and swims away as a teletroch. The teletroch finds a new substrate. It attaches. The temporary cilia disappear. The organism secretes stalk material, which solidifies into the new stalk. Within hours, it metamorphoses into a sessile adult. The whole process takes 20 to 30 minutes. Under ideal conditions, Vorticella populations explode. Every 3 to 4 days, each individual divides. 1 becomes 2, 2 become 4, 4 become 8. Exponential growth fills the environment with bell-shaped organisms clustered on every surface. When conditions worsen, Vorticella detaches from its stalk and swims freely using the aboral cilia band. Or it forms a protective cyst. The cyst can survive drying and other stresses. When water returns, the cyst ruptures and the organism emerges. Sexual reproduction happens by conjugation. Small free-swimming microconjugates find large sessile macroconjugates. The two fuse temporarily and exchange micronuclear material. After separation, both undergo complex nuclear reorganization, producing genetically diverse offspring. Vorticella has been found fossilized in rocks 250 million years old. Triassic period. The fossils show spirally contracting stalks identical to modern species. Form and function have barely changed across geological time. Scientists use Vorticella in microfluidic research. The feeding current creates flows useful for mixing samples. The contractile stalk inspires biomimetic actuators. Engineers are designing microscale robots based on spasmoneme mechanics. The calcium-responsive protein fibers could become artificial muscles for soft robotics. Vorticella lives in fresh water primarily, but some species tolerate brackish water. They cluster on vegetation, creating visible fuzz. In aquarium hobby, people sometimes see Vorticella growing on shrimp or fish. It looks like white fungus, but it is not fungus. It is ciliates, usually harmless unless populations grow so large they clog gills. Then they suffocate the host. Most cases stay benign. More than 200 Vorticella species exist. They differ in size, color, and minor structural details. Telling them apart requires electron microscopy and genomic analysis. Under light microscopy, they all look like bells on stalks. The macronucleus appears as a curved band shaped like the letter C. The micronucleus sits nearby as a small round structure. Contractile vacuoles pulse rhythmically, collecting excess water and expelling it. The pellicle shows striations running parallel around the cell. These are ridges and grooves in the surface. Some species have pustules or warty projections on the pellicle. Others are smooth. Bacteria sometimes grow on Vorticella bodies as epibionts. This can look like part of the organism, but it is not. The bacteria are separate species hitching a ride. Some relationships may be symbiotic. The bacteria produce compounds that help the Vorticella. Evidence remains incomplete. Rotifers eat Vorticella, so do some ciliates. Predation keeps populations in check. Without it, Vorticella would carpet every surface. The balance maintains ecosystem function. Ponds need Vorticella to control bacterial populations. Vorticella needs bacteria for food. Bacteria need organic matter to decompose. Everything connects. One species disappears and the web shifts. Vorticella simplicity makes it perfect for teaching. Students can easily observe the feeding current, the contractile stalk, the nuclear structure, and the reproductive process. Culturing is simple. Put pond water, some rice grains for bacteria, and algae for substrate in a jar. Vorticella appear within weeks. Subculture every 2 to 3 weeks for continuous supply. No special equipment needed beyond a basic microscope. No 2 moments look the same. Amoeba proteus measures 250 to 600 micrometers in maximum diameter, large enough to see with naked eyes as a tiny whitish blob. Under microscopy, it appears as colorless, translucent jelly with a dark nucleus inside. The cell has 2 cytoplasm layers. Outer ectoplasm forms a stiff gel layer. Inner endoplasm remains fluid. The membrane can flex freely because amoebas have no cell wall. This allows movement through pure flow. Pseudopods form when pressure from ectoplasm forces endoplasm forward. The membrane bulges. Endoplasm streams into the bulge. When it reaches the tip of the membrane, the pressure causes more bulging. The pseudopod extends. At the rear, adhesions release and the trailing edge retracts. Net result, the amoeba flows forward. Scientists call this amoeboid movement. It is one of the most primitive forms of locomotion, slow, around 1 micrometer per second, but effective in 3-dimensional environments. Amoeba proteus lives on the bottom of freshwater ponds and streams, especially in decaying vegetation. It eats bacteria, algae, rotifers, and even smaller amoebas. When prey comes near the amoeba, it extends pseudopods that surround the food. The pseudopods meet on the far side. The membrane fuses. Now the prey sits inside a food vacuole suspended in the amoeba's cytoplasm. Digestive enzymes flood into the vacuole. The prey dies and dissolves. The amoeba absorbs nutrients. Undigested waste stays in the vacuole until the membrane fuses with the cell surface and dumps it outside. This process is phagocytosis. Cell eating, the amoeba has no mouth, no fixed spot for ingestion. Phagocytosis happens anywhere on the surface. Studies show prey do not detect the slow-moving amoeba. They keep swimming or crawling until pseudopods surround them. By the time they realize danger, the trap is closed. Inside the endoplasm floats one large nucleus. It contains the genetic material. DNA organized in chromosomes. Multiple mitochondria generate ATP. Golgi apparatus processes proteins. Food vacuoles at various stages of digestion. A contractile vacuole collects excess water. Amoeba proteus lives in freshwater. Osmotic pressure constantly forces water into the cell. Without the contractile vacuole, the cell would swell and burst. The vacuole fills over minutes, then contracts, expelling water through a pore in the membrane. The cycle repeats constantly. Amoeba reproduces by binary fission. Asexual reproduction. The nucleus divides first, then the cell pinches in 2. Each daughter cell gets 1 nucleus. They enlarge to parent size within hours. The entire process takes less than 60 minutes. Before division, the amoeba rounds up into a ball. Pseudopods retract. Over 15 minutes it splits. When conditions turn bad, amoeba forms a cyst. It loses most of its water, secretes a protective coat made of chitin-like material. Inside the cyst, mitosis happens multiple times. When the cyst ruptures, several daughter amoebas emerge. This is survival reproduction. It creates backups. Some amoeba species reproduce sexually under extreme stress. Spores form, gametes fuse. This introduces genetic variation. but sexual reproduction remains rare and poorly documented in Amoeba proteus. Amoeba proteus lacks true symmetry, no anterior or posterior, no dorsal or ventral, just dynamic flow. This makes studying it challenging, no fixed landmarks. The nucleus moves throughout the cytoplasm. Organelles drift, everything flows. Scientists use molecular markers and fluorescent proteins to track structures. Erasmus Darwin, in 1803, recorded an experiment where he desiccated amoebas and later rehydrated them. They survived. This suggests early researchers knew about cyst formation and cryptobiosis even in amoeba. The ability protects them when ponds dry. Research shows amoeba movement follows low-dimensional chaotic dynamics. The correlation dimension sits around 3 to 4. That means the seemingly random pseudopod extensions arise from deterministic interactions between a small number of processes. Sol-gel transformations in the cytoplasm, cytoplasmic streaming, calcium-mediated reactions. These cooperate to produce complex behavior from simple rules. Amoeba proteus has most proteins associated with sexual reproduction. The machinery exists. Meiosis genes, recombination genes. But scientists have never observed actual sexual reproduction or meiosis in laboratory cultures. Either it happens extremely rarely, or the genes serve other functions. Amoeba lives worldwide, every continent except Antarctica. Any freshwater body with decaying vegetation probably contains amoeba. They play important roles in microbial food webs. Eating bacteria controls bacterial populations. Being eaten by rotifers and small crustaceans transfers energy up the food chain. Some amoeba species host pathogenic bacteria, Legionella. The cause of Legionnaire's disease can survive inside amoeba. The amoeba protects the bacteria from environmental stress. When amoeba gets eaten by humans through contaminated water, the Legionella infects us. Amoeba acts as a reservoir and vector. Other amoeba species cause disease directly. Naegleria fowleri. The brain-eating amoeba enters through the nose when people swim in warm, fresh water. It migrates to the brain and causes primary amoebic meningoencephalitis, nearly always fatal. Entamoeba histolytica causes amoebic dysentery, spread through contaminated water and unwashed food, settles in the intestines, eats tissue, causes severe diarrhea. But Amoeba proteus itself is harmless. It lives in pond water. It eats microscopic organisms. It has no interest in larger animals. Classrooms use Amoeba proteus for teaching cell biology. Easy to culture, easy to observe. Shows all basic cellular processes. Movement, feeding, reproduction. Students can watch living cells behave in real time. No fixation, no staining. Just drop pond water on a slide and look. The 7 organisms in this list share something. They survive by breaking rules. Stentor contracts to 1/6 its size using molecular springs. Paramecium computes escape routes with one cell. Tardigrades survive space. Bdelloids reproduce without sex for 25 million years. Hydra regenerates from cellular soup. Vorticella builds engines more powerful than cars. Amoeba flows like liquid but hunts like a predator. Go outside. Find a pond. Any pond. Collect one drop. Mount it on a slide. Look through a microscope. Somewhere in that drop swims at least one of these organisms, probably several. They have been there for hundreds of millions of years. They will be there long after we are gone, because they mastered survival at the smallest scale. And that scale does not care about mass extinctions, climate change, or cosmic radiation. It just keeps flowing, dividing, regenerating, forever.